Hi, I'm Sean Mooney, Professor and Chief Research Information Officer of UW Medicine at the University of Washington. In this video, Professor Anant Madabhushi from Case Western Reserve University will present a talk titled Artificial Intelligence, Radiomics and Pathomics, Implications for Precision Medicine. Please be sure to click the link below to register for our mailing list to be alerted for upcoming talks and videos. We hope you enjoy these.
All right, so welcome everyone to the AI and Data Science in Medicine seminar. We're really excited to kick off the seminar series today with our first external speaker. I'm Hannah, I'm a PhD student in Biomedical and Health Informatics here at UW, and I have the pleasure of introducing our speaker today. But first we'll have Sean Mooney say a few words for us.
Great, thank you, Hannah. Welcome everyone. I just want to confirm, did Anant rejoin? We just did a technical check for him a few minutes ago and I haven't seen him.
I'm on.
You're there?
Yeah.
Oh yeah, okay, great.
Yeah, yeah, I just need to change my name here. Sorry, that was my son.
No worries. So welcome everyone. Really excited for you to all join us. We're also excited about announcing this is the first external seminar that we've done for this UW AI and Data Science in Medicine series. And so first of all, I want to thank everyone for registering their interest in the REDCap forum. Again, reminder, you only need to do that once, but we'd really like to be able to capture your contact info so we can give you updated Zoom links and also remind you about the seminars that are coming out. And if you're getting too many emails from me, just let me know and I'll unsubscribe you manually. As a reminder, if your department has relevant seminars or other events that you'd like to let us know about, please let me know and I'll add them to those mailings. If you look at the bottom of each of those emails that goes out, they have a list of other seminars or other workshops and conferences and things like that that are, worth being aware of as we go forward. So there's a lot more to come. So again, before we kick off today, really excited also to talk about our upcoming seminars. Barbara Young, Chair of Medicine at UW Medicine, and Stephen Finn, Head of the Division of Internal Medicine at UW, will be presenting our next seminar on December 7th, which will be an internal one. And then we have a really awesome lineup of external speakers that are coming up over the next 3 or 4 months, with Zach Kahane presenting in December 14th. He's Chair of Biomedical Informatics at Harvard. Keith Dreyer, who's the Chief Data Science Officer at Mass General, also a faculty member at Harvard, and Mia Levy, the Director of the Rush University Cancer Center, will be presenting in December. So that's December, January, and February. But today we're really excited about Anant, and I'm going to turn it back over to Hannah, who will do a very quick intro.
Thanks, John. So now I'm pleased to introduce Dr. Anant Madabhushi, who is a professor of biomedical engineering at Case Western Reserve University and a research scientist at the Louis Stokes Cleveland VA Medical Center. He authored more than 400 peer-reviewed journal articles and conference papers, has gained international attention in the field of biomedical engineering, and earned numerous awards and recognitions in academia, as well as mainstream media. We're really lucky and honored to have him here today. So Anant, if you're ready, take it away.
Thank you, Hannah. Thank you, Sean, for that kind introduction. I'll go ahead and share my screen, so hopefully you guys can see it. Yep.
Yes, we can. Thank you.
Great. Thank you. Thanks. So just before I get started, I just was pleasantly surprised to find that Dr. Satish Chandratre is on. He actually was one of my first bosses back when I was still a graduate student undergraduate student, actually, in India. So just a shout out to my old mentor. Thank you for making this. I decided to spice up the title a little bit. I realized that I've been using the same title for a while, and I was really inspired by a book which I'm sure a lot of you on this call are familiar with, The Emperor of Maladies by Siddhartha Mukherjee. And so as I think about what we do, or at least the core crux of my talk today, which is around AI for precision medicine, I can't help but think of artificial intelligence really being the interpreter of all maladies. And so just acknowledgement of that wonderful book by Siddhartha Mukherjee. I should just mention, of course, that as director of the Center for Computational Imaging and Personalized Diagnostics, I have the real pleasure of working with, really more than mentoring, just working with some really amazing individuals, trainees, PhD students, postdocs, faculty. And so really what I'm showing today is really a reflection of the amazing work that I get to be a part of. And also just a shout out to the Louis Stokes Cleveland VA, where I'm a research health scientist, and we're working on using AI to really help improve the health of our veterans. As the saying goes, no conflict, no interest. So I guess it's a good thing that we are working with industry to move the needle forward, in a manner of speaking, taking the discoveries and advances that we're making in the lab and moving that forward from the basic discovery to the bedside. I guess my true disclosure, though, is the fact that I also have a social media presence. So quite unabashedly, unashamedly, I'm advertising here my Twitter handle. If any of you are on social media and any of what I talk today is remotely appealing, please feel free to give me a follow. All right, so I want to contextualize the problem today by first talking about cancer. I will talk about non-oncologic conditions as well, but I think cancer is a really good disease system in the sense that it really conveys the scope of the problem in many ways for management and treatment of a number of diseases, and the potential role for AI to really help in addressing this issue of precision medicine. So if you look at the statistics today, about 50% of all men in the United States will be diagnosed with some form of cancer in their lifetime. Now, for women, it's only marginally better. 1 in 3 women will be diagnosed with some form of cancer in our lifetime, which means that effectively 40% of the US population will be diagnosed with some form of cancer in his or her lifetime. And when you look at the actual mortality on account of cancer, these are still pretty staggering statistics. About 600,000 deaths on account of cancer in a year. Now, while these are certainly very high numbers, large numbers, there's a little bit of a discordance and discrepancy between a diagnostic incidence rate of about 40% of men and women in the US being diagnosed with cancer versus a statistic of about 600,000 deaths because of cancer in a year. And the reason for that, of course, is the fact that we've become much better at early identification of a number of cancers. We've got better biomarkers that we use for screening for a number of cancers. We're using imaging more effectively, more aggressively to find cancers at an earlier stage. And we know that the earlier that you find these cancers, the better the mortality related likelihood or the better the chances of survival. And of course, we also have better therapies today. Immunotherapy, which I'm sure everybody's aware of, which got the Nobel Prize for Medicine in 2018, has really changed the landscape of the treatment for cancers. But if we are completely honest, we have to acknowledge that part of the reason why there is a discrepancy between this very high diagnostic incidence rate or detection rate and the mortality, in part, at least in part, is due to the fact that there is overdiagnosis and overtreatment for a number of cancers. And perhaps nowhere is this better exemplified than in the context of prostate cancer, a disease that affects 1 in 6, 1 in 7 men in the United States. And yet only 1 in 40 will actually die on account of it. In fact, men are more likely to die with prostate cancer rather than on account of prostate cancer. And so this is a classic case in point where most times the cancer that's diagnosed is relatively indolent, it's not aggressive, and yet, certainly in the United States, we tend to throw the kitchen sink at it. In other words, most men who are diagnosed with prostate cancer in the United States will get radical therapy, most likely a radical prostatectomy. It's an issue that is really affecting the health and the outcome for a number of these men, because we know that there are side effects that are associated with a lot of these radical interventions. And it's not just the patient-centric toxicity that overtreatment brings to the table. There is also the very real issue of financial toxicity. I mean, these are some staggering statistics that came out a couple of years ago, showing that about 42% of new cancer patients will lose their life savings. About 62% of cancer patients report being in debt due to their treatment, many of them within 1 year of that initial diagnosis. So we have to acknowledge that overtreatment and overdiagnosis of a number of these cancers not only has direct deleterious effects to the patient, but it also has the unfortunate consequence of financial toxicity and economic implications and financial implications to the patients and of course, to the larger healthcare system. And so that's really the preamble for what I want to talk about today, about the role for artificial intelligence, not just in the context of disease diagnosis, which is where, of course, we're hearing a lot of the buzz right now. A lot of the articles we see in the magazines, in the press, a lot of the new FDA approvals, are really in the diagnostic setting. And like I said before, I mean, this is important. We need AI for disease detection and diagnosis. But again, if you go back to that number, 40% of the adult population will be diagnosed with some form of cancer. We're really left with this issue of how do we deal with the management and the treatment of these cancer patients after that diagnosis. And so what I really want to spend most of my time talking about today is, now, how can AI really move the needle forward in terms of better prognostication of disease outcome? How can AI be used in prediction of therapeutic response, in being able to identify which individuals will benefit from additional therapy, more aggressive therapy? Because at the end of the day, it's really the development and the application of these prognostic and predictive tools that really will allow for tailoring the right therapy for a given patient based on their specific risk profile. And ultimately, that's really the realization of this promise of precision medicine that we all seek. Now, one of the things that we have begun to appreciate and understand and are doing on a fairly routine basis is multi-scale disease characterization. So even this is not really in the research realm anymore. For a number of diseases, a number of cancers, we are routinely acquiring data that allows for characterization of the disease across multiple different length scales. So as an example, let's look at prostate cancer, where in the context of an individual who has adenocarcinoma, chances are pretty high now that they will get a multiparametric MRI exam. So what you're seeing here is an area of adenocarcinoma on an endorectal T2 weighted MRI scan. But with a multiparametric sequence, you can also start to look at other parameters. You can look at diffusion weighted imaging, where you look at water mobility in the tumor. You can look at choline, creatine, citrate, and metabolic concentrations. You can look at the vascular attributes of the tumor through dynamic contrast enhanced perfusion imaging. Now, assuming that this patient goes on to get a radical prostatectomy, with the advent of whole slide digital imaging, you can digitize the pathology specimen and start to analyze and interrogate that same region of tumor that I showed you on the MRI scan with the computational tools at a highly granular microscopic resolution. In other words, you can start to look at the spatial architecture and spatial arrangement of individual cells. You can look at the textural attributes of the different tissue compartments, look at the textual attributes of the stroma, the epithelium, and then go further down in the length scale. You can start to look at the shape of individual nuclei. And you could go even further, as is happening fairly routinely for a number of cancers, like I mentioned, where some sort of genomics or proteomics is being acquired. And for instance, you can now with laser capture microdissection, you can go in specifically into that region that I showed you, on the MRI scan, look at that corresponding region on the pathology and start to do, for instance, mass spec and get a proteomic signature. So you've got information for this one patient that provides characterization across multiple length scales, from radiology to pathology down to the molecular proteomic level. And the question that we've been asking at the Center for Computational Imaging and Personalized Diagnostics at Case Western is, how do we take advantage of the plurality of this information? How can we start to integrate, combine all of this information across different length scales? What are the kinds of features? What are the kinds of subvisual features that may not be visually discernible by a radiologist or a pathologist? What are those attributes that we can start to extract? And more critically, how can we start to combine the plurality of that data across these different length scales to be able to create integrated predictors for not just to improve diagnosis, but also being able to prognosticate outcome, to prognosticate risk, to prognosticate progression, and critically, from a clinician standpoint, being able to predict therapeutic response. Now, I want to quickly add that this concept of a companion diagnostic, this idea of using tools to prognosticate cancer outcome or prognosticate which therapy is appropriate for a given patient, is not a new idea. So I just want to put that out there. It's not like people have not been doing this. In fact, perhaps the most famous companion diagnostic test that's out there is the one that's marketed by Genomic Health, now part of Exact Biosciences, Oncotype DX, a 21 gene expression assay that essentially allows for identifying which early stage ER-positive breast cancer patients will benefit from adjuvant chemotherapy. The reason this test became very popular is because over 150,000 women in the US would be diagnosed with early stage ER-positive breast cancer, and yet the vast majority of them, over 100,000 of these women, will have not very aggressive disease. In other words, they will respond to hormonal therapy and surgery alone, in which case they don't need additional chemotherapy. And so the test came along because really it was a way of identifying which of these women identified with early stage ER-positive breast cancer could avoid adjuvant chemotherapy and therefore not have to deal with all the deleterious side effects of chemotherapy, because quite simply they didn't get any added benefit. They didn't need the adjuvant chemotherapy. The problem, however, is this is an expensive test. It's also tissue destructive. It involves destroying the tissue in order to do the sequencing and the gene expression profiling. But also, importantly, we know that tumors tend to be extremely heterogeneous. We know about the fact that the more aggressive tumors tend to have multiple subclones, and the presence of multiclonality, multiple different clones within the tumor, makes these very difficult to treat. And so when you sample the tumor to do the gene expression profiling, to do the molecular profiling, unless you've really captured the most aggressive component of the tumor, the corresponding risk score or risk assessment that the companion diagnostic comes back with may not be a truly accurate reflection of the aggressiveness potential of the tumor. Like I mentioned, with the advent of whole slide imaging, we now are routinely acquiring and scanning digital pathology slide images. So this is an example of a standard H&E slide, which is pretty much sort of the go-to slide and has been for over 100 years for pathologists for the diagnosis of diseases, like cancer. But with the advent of whole slide imaging, you can scan these H&E slides into very high resolution images like the one that you see here. And AI tools and computational pathology tools can be trained to go in and identify individual cells, individual cancer nuclei, individual lymphocytes. And you can start to look at the spatial architecture and the spatial arrangement of these individual cells and individual nuclei using techniques from network theory and computer science. Like I said, you can start to apply mathematical operators like texture features to look at the heterogeneity within the individual tissue compartments, look at the heterogeneity within the stroma, within the epithelium, even within individual nuclei, and convert the entire landscape of this tumor into a digital signature or a digital biomarker. And the beauty is it's completely non-destructive. You're just working off an image of a slide, and that provides a quantitative, reproducible digital biomarker that provides a characterization of the tumor morphology in a way that's unprecedented, in a way that simply cannot be done by visual assessment of the pathology slide alone. And the reason I got really excited about this concept was because 13 or 14 years ago, when I first started working in computational pathology or application of machine learning to digital pathology images, I started to focus on problems that were very near and dear to pathologists. So problems around disease detection and grading, right? So pathologists try to grade the severity of the disease, and they typically tend to have very fixed lexicons on how you do grading of a variety of cancers, be it breast or be it prostate cancer. And so that's what a lot of my early work was on. And that changed in 2008, 2009, when I had the good fortune of meeting Sridhar Ganesan, a breast cancer oncologist at Cancer Institute of New Jersey. And Sridhar saw the work I was doing and said, look, Anant, everything they're doing is great, but as a clinician, it doesn't benefit me. What I want to know is not just the cancer grade. I want to know which women should I treat with chemotherapy and which women can avoid chemotherapy. That's my pain point. And can you help me with that? And so that's when we started to brainstorm, and we came up with a concept of the image-based risk score, which in a sense was similar to the concept of Oncotype and the risk score that Oncotype had, or the Genomic Health had. The difference being that this risk score was solely coming off the computational interrogation of the morphology of the tumor from an H&E slide image. So, every woman suspected of breast cancer will get a biopsy. So there'll be tissue available, tissue slides that are analyzed by the pathologist. And all we were doing was digitizing that glass slide and then subjecting it to the computational analysis and the image algorithms. And by identifying patterns of disease aggressiveness, like I mentioned, spatial arrangement of cells and nuclei, and we'll talk a little bit about this, we were able to come up with a risk assessment of, is this more severe disease, in which case they needed the chemotherapy, or is this less severe disease, in which case they could possibly avoid the more aggressive treatments. Well, how do you go about building this? Well, one of the things that we realized we needed to do was to understand the tumor morphology. Well, we had to go in and identify the individual cells. We had to find the individual structures or primitives in the pathology image. And that's where deep learning has been such a boom, right? So we hear about deep learning, pretty much everywhere. Like, how can you not hear about deep learning, right? But deep learning has really started to have a significant impact in the area of pathology, because pathology, in fact, perhaps even more so than radiology, is really very nicely set up for problems in computational pathology, just simply because of the size of these images, because these are large images, and therefore these data-hungry algorithms, which require a large amount of training data that they ingest, it's very opportune for developing these deep learning, machine learning algorithms that can learn the best representations to distinguish between the categories of interest. And so the beauty is, in some ways, is that these architectures, these neural network architectures that comprise deep learning, essentially involve providing as input annotated examples of these images. These images then go into the network. You've identified or labeled a bunch of these training exemplars, and then the network learns the best representation to separate out the category of interest from everything else, and then spits out the output. And so this was a paper we published a few years ago where we showed one of the first examples of deep learning applied for cell detection in pathology images. We showed that simply by standing up one of these networks, by providing it a large number of well-annotated training exemplars, you could actually get a result that looked like this, where essentially on a cell-by-cell basis, you had a segmentation, you had a result that popped out. A couple of years ago, we said, well, we've shown that this works in cell detection, cell segmentation. Could we up the ante here? Could we potentially challenge the deep learning to ask or address, more challenging questions that really involve higher level predictions at a patient level? And so, working with Ken Margelis, Mike Feldman, and Preeti Lal at the University of Pennsylvania, we came up with this idea of trying to look at the ability of deep learning to predict heart failure from endomyocardial biopsies. So essentially, this was a project which involved getting a series of patient samples, biopsies from Penn. These were digitized at Penn on a whole slide scanner. And we got a set of about 100 patients, half of whom had failing hearts and the other, where the biopsies were normal. And we used that data to train a neural network deep learning algorithm to identify whether these patients had evidence of heart failure or not. Then we got another set of 100 patients that we used as a validation set, scanned at the same time. And that served as a basis for validating how the algorithm did. And just to make it interesting, we also had two pathologists analyzed the validation set. And so they had the same question that was posed of the algorithm was posed to them. Can you identify features of failing hearts versus biopsies that didn't have any evidence of failure? And so when we did a head-to-head comparison, the neural network came out with an area under the curve of 97%. On the other hand, two pathologists had an average area under the curve of 74%. So there was about a 24% differential between what the machine was able to do and what the pathologists were able to do. So we were thrilled to bits. We put out a press release. GE Reports picked it up, calling it one of the most exciting things on the planet that week. I was briefly trending on Reddit, so it was great. It was all well and good. However, the postscript of the story was that a few weeks later, we asked Penn to send us another tranche of cases. So they said, that's fine. So they found another cohort of 90 patients, again, to serve as a second validation set. They digitized it on the same scanner and sent it to us. And we ran the same algorithm on that set of patients. And lo and behold, the algorithm went from 97% down to 75%. And this was a real, this really stumped us because we could not understand how patients from the same institution, basically the same demographics, the same slide scanner, the same lab, where really there shouldn't have been any variation at all. How could the network that had already been validated on one tranche perform so badly or relatively badly on the second tranche? Thankfully, like I said at the start, I have the privilege of working with really smart trainees, and they did the digging and then found out that the thing that had changed between the second tranche and the third tranche of cases we got was that a remote software upgrade had been applied to the digital slide scanner, and that had changed or perturbed the distributions of these images ever so slightly. And that had really thrown off the network. That's why it went from 97% down to about 75%. And so this is one of the challenges, right? And so the message here is that we need to be aware of the fact that these deep learning algorithms have, this issue of transparency. There is an opacity associated with these representations, and that was sort of a real, enlightening study for us, because we realized that when you don't understand what the inner working of the algorithm is, then when something breaks down, sometimes it can be really tricky, really challenging to figure out, what went wrong. And this is, a sentiment that's been echoed by several others. I mean, this is Professor Jordan at UC Berkeley, who said, trusting these brute force algorithms too much is a faith misplaced. And Cynthia Rudin, at Duke University, wrote an amazing paper in Nature Machine Intelligence last year, saying, we have to stop trusting these black boxes. And this is another great example from Dr. Samir Singh at UC Irvine a couple of years ago, where he and a student were trying to train a neural network to distinguish huskies from wolves. I mentioned this example several times, but it's worth repeating because I think it really reinforces the fact that when you don't understand how the black box works, you can't really understand the interpretations. You don't know where these interpretations are coming from. You can't predict what the interpretations might look like. And so Samir, working with his student, tried to train a network to distinguish huskies from wolves. They got a bunch of images from Google, trained the network, and Samir at the onset knew that this was going to be a tough problem because huskies and wolves look very similar to each other. But when the network came back on the test set and reported a 99% accuracy, Samir, who's clearly way smarter than I am because he immediately challenged the results, immediately knew that something was off, went in with his student and tried to figure out what had gone wrong. And what they discovered was all the images of huskies had some white on the background. There was always some snow. And that was what was throwing off that was what the algorithm was picking up. It wasn't actually features of the face of the animal. It was picking up the fact that huskies were always against some snow in the background. And that's what the network was picking up. And so that sort of further reinforces this issue that we have to be very aware of what these networks are learning, right? However, deep learning algorithms can be very useful. So I will concede, sorry, there's a little election joke there. I will concede that deep learning networks can be extremely useful for segmentation of histologic primitives and structures. So for instance, we showed a couple of years ago that you could train these networks to identify a series of different kinds of primitives. You could identify mitotic figures, cells, lymphocytes. You could segment out different tissue compartments, stroma from epithelium. We've now shown that these networks can be very, very powerful, running on very, very large images to segment hundreds of thousands of immune cells, lymphocytes, cancer cells, really in pretty much close to real-time efficiency. And not only is it the fact that they can segment out these primitives very, very quickly and very efficiently, you can also train up your own networks in a pretty efficient fashion. So this is work that's being developed in our group. There are other groups that are pursuing similar efforts, where you can stand up these deep learning networks pretty rapidly. One of the challenges hitherto has been that these, because they're so data hungry, these neural networks are so data hungry, you're going to provide them hundreds, thousands of images which are deeply annotated. Well, who has the bandwidth to do all those annotations? So we developed something called Quick Annotator, which we hope to make publicly available very, very soon, which essentially allows for the end user to annotate just a few examples of the region of interest, in this case, cells. The network takes just a few examples, generates an output. The end user then, in active learning mode, is able to edit out the results of that segmentation, passes it back to the neural network that uses the updated results to refine its performance. And then by iterating back and forth, the neural network is able to converge to a pretty good result very, very quickly, as opposed to having to segment out hundreds of thousands of images at the get-go. So we call this Quick Annotator. Like I said, we're hoping to make this available to the community in the near future. The other thing that, of course, these networks are really good at doing is that they allow for classifications of individual cells. You can classify lymphocytes from cancer nuclei. You can look at macrophages, fibroblasts. You can also segment out, like I said, stroma from epithelium, and really look at the organization, the architecture of the different cell types within different tissue compartments, with a net result that we can now start to translate all of this into digital biomarkers that can allow us to predict outcome and predict therapeutic response. So you could see that sort of the theme I'm really getting at here is that you've got the opaque representations with deep learning, but the efforts in our group has been more focused around interpretable features, intuitive features, organic features, features that are really connected to the domain or the pathobiology of the disease. And here's one example of one of those features. We've known for a long time that in pathology, collagen fibers and collagen fiber orientation clearly has a lot of value, clearly is extremely important. And so we undertook this study to try to figure out from individual H&E images alone, could we identify the orientation architecture of collagen fibers and use features relating to the architecture and organization of the collagen to be able to then prognosticate outcomes. So we did a study in breast cancer patients where we had short-term survivors and long-term survivors. We trained the networks to go in, segment out the tumor, but also segment out, you can see in blue here, the collagen fibers. And essentially, we were able to develop a feature, a biomarker, if you will, that captured the orientation or the organization of the collagen fibers with respect to the tumor edge. And what we found was that in patients who tended to do poorly, the collagen vectors, the vectors corresponding to collagen fibers, were all oriented in the same direction. However, in patients who did well, that is, their overall long-term prognosis was good, the vectors were more haphazardly oriented, more haphazardly arranged. And while that might seem counterintuitive, it actually makes sense when you think about the fact that the collagen fibers potentially provide a highway or expressway for the cancer cells to hitch a ride on. And so if you think about it, the more organized, the better your highway, the easier it is for a cancer cell to migrate from the primary site to distant sites. And essentially, that helps promote metastasis and promotes progression. On the other hand, if it's more difficult to move along that highway, if the highway is impeded or tortuous, it's more difficult to hitch a ride. and therefore you're limited largely to the primary site. And so that's what we've observed with this collagen fiber orientation, which we've now trained with a machine classifier in a paper that's currently under review. We've demonstrated in over 400 patients that this collagen fiber orientation alone is a biomarker of survival in early stage ER-positive breast cancer patients. And so it's an example of one of those features that's connected to the pathobiology of the domain of the disease. We know it has clear interpretability, it has clear intuitiveness, we understand where it comes from, and best of all, of course, we've demonstrated pretty comprehensively that it is prognostic of survival. But it's not just, it's not just collagen fibers. I mean, there are other simpler things almost that we can identify. Features, for instance, relating to the shape and the size and the arrangement of nuclei, cancer nuclei from breast cancer images. The formation of tubules. We can train networks to go in and find the number of tubules in breast pathology images. We can identify mitotic count. Now, a number of papers have been published on identifying mitotic figures. What we've done, essentially taken all of the features relating to nuclear morphology, tubules, mitotic count, created a multi-factor, multi-variable classifier that ingests all of these features relating to these different attributes that pathologists, by the way, know and they understand. They understand these attributes. But by quantifying it, by having a computational assessment of these hallmarks of the cancer, we're able to now come up with a prognostic predictor that we demonstrate is not just prognostic in early stage disease or lymph node negative breast cancers, but is also prognostic in lymph node positive breast cancers. So these are the Kaplan-Meier curves. And to those who might not be that familiar with these curves, these are survival curves. And so if you see a significant separation between the blue line and the red line, that means your classifier is able to separate out high risk and low risk patients. That means your classifier is doing what it should be doing. That is, it's an effective classifier. And so what we've demonstrated now in data from multiple different sites, including patients from India, from the Tata Cancer Center in Mumbai, that we have the ability to be able to prognosticate outcomes, survival, both in lymph node negative and lymph node positive patients. And it's not some opaque black box, but actually using hallmarks and features that are computationally derived, but link back, like I said, to the pathobiology of the disease. And so why is all of this important? Well, just a head-to-head comparison. It's a little bit tongue-in-cheek, so you'll indulge me here. I talked about Oncotype DX. Like I said, it's an expensive test, $4,600. If you think about the IBRIS test, well, it's pennies on the dollar. It's all it really costs is the electricity and the computational resources needed to run the algorithms. It's a rapid test because with this kind of analysis, you can return the test back to the ordering clinician anywhere in the world. You don't physically ship tissue like with Oncotype DX. You don't destroy any tissue. It's all digital image analysis. You can have a global impact as opposed to largely being limited to the US alone because of the necessity with a molecular test for physically shipping tissue. So in other words, you have an inexpensive, fast, reliable, accessible prognostic breast cancer test that's priceless for everything else that's MasterCard. All right, what else? Well, it turns out that you can also combine IBRIS with Oncotype DX. So we've shown in a study a couple of years ago in about 378 patients that when you combined the Oncotype DX with the image-based risk score, you could significantly improve the performance of Oncotype, nearly by 20%. So in other words, you can identify 20% more truly low-risk breast cancer patients who could successfully avoid adjuvant chemotherapy. We've also shown now in a paper that we published last year that when you look at ductal carcinoma in situ, that you can better prognosticate which ductal carcinoma in situ or stage 0 breast cancer as it's called, which of these stage 0 breast cancers will progress on to invasive ductal carcinoma versus those DCIs that will not. And so we demonstrated both with a training set as well as an independent validation set that the features of the nuclei, the features that I alluded to before, arrangement, shape, orientation, disorder, and texture, were prognostic of progression from pre-malignancy or DCIs to stage 0 to invasive ductal carcinoma. In prostate cancer, one of the big questions is, even in men who undergo surgery, one of the big questions is which of these men will end up having a recurrence of the cancer. In which of these men will the cancer come back? And so we did a study which is currently under review, where we looked at features relating to the shape and the orientation of individual glands on the surgically resected specimens. And we trained a classifier. This is a study with almost 1,000 patients from 6 different sites. We used one of the sites for training the classifier and used a large cohort of almost 600 patients to do the independent validation, and found that there was a clear separation between those patients who did and did not have cancer recurrence. What perhaps was even more satisfying was the fact that when we looked at margin negative patients, in other words, patients who had truly low risk disease because they were margin negative, even there, we were able to identify a set of patients who had worse outcome. And so suggesting that there may be another subset of patients who could benefit from adjuvant therapies, say, for instance, docetaxel in the adjuvant setting or radiotherapy in the adjuvant setting. I talked about Oncotype DX for breast cancer. Well, it turns out there is a molecular test for prostate cancer to predict risk of metastasis after surgery called the Decipher test. Like Oncotype, it's a 22 gene expression based assay. Like Oncotype, it's tissue destructive. Like Oncotype, it's expensive. It's over $3,000, but it's in the NCCN guidelines and it's used for prognosticating risk after surgery. And so we, in the study, did a head-to-head comparison. We went head-to-head with the Decipher test on 175 patients and said, okay, let's try to figure out, when you look at the image-based risk score in Decipher, is there any improvement? How much better is Decipher? And what was nice was we found equivalence. We found non-inferiority. We found that the image score and the Decipher were almost equivalent, but it gets better. What we then did was we took the image score, we added PSA, the preoperative specific antigen, which is the prostate specific antigen, and as well as the Gleason score, standard routine clinical variables that you're getting for all these prostate cancer patients. We took those, combined it with the image score, and actually showed that you could get a performance that was nearly twice as good as the Decipher, all the while not destroying any tissue, all the while doing it really for pennies on the dollar. And so not only were we able to show non-inferiority, by coupling the image score with a couple of standard clinical parameters, our initial data, now this still needs to be validated with a larger set of patients, but our initial data on 177 patients seems to be really promising, because it seems to suggest that we can get a hazard ratio that's nearly double what we're getting with the Decipher in prognosticating cancer recurrence risk after surgery. One of the things that obviously in this time of COVID and the pandemic, we're all too familiar with the cytokine response and the strong immune response that the virus solicits. But we know that having an immune response when it comes to cancer is actually not a bad thing. It tends to confer a certain prognostic advantage for a number of cancers. There's been a lot of work done around trying to quantify the density of the immune response as a way of prognosticating better outcome versus poorer outcome for a number of cancers. In a paper that we published last year in Clinical Cancer Research, we took a slightly different tack. Rather than solely focusing on density of the immune response, we decided to look at the spatial arrangement of the immune response. The question we were asking is, if you look at the interplay between the immune cells and the cancer cells, is there something unique that emerges? Is there something about the spatial co-arrangement of cancer cells and immune cells that could actually tell us about which of these patients is going to have recurrence versus not? And we studied this in the context of lung cancer. And so we trained our machine learning to go in and identify immune cells, and you can see that these in the green cluster here, and also the cancer cells, and so this is in the red cluster. What we found was that in patients who did not have recurrence, there was almost this enveloping or mothering effect that the immune cells conferred in these tumors and surrounded the cancer cluster, so almost preventing the cancer cluster from breaking out and branching out. On the other hand, when we looked at patients who did have recurrence, there was a much larger cancer cluster and a much smaller immune cluster. So it didn't really have that mothering or enveloping effect or protective effect around the cancer nuclear cluster. And we actually showed that this particular spatial arrangement of immune cells and cancer cells was strongly prognostic of recurrence in early stage lung cancer. We've taken that approach and now demonstrated across different indications that the spatial arrangement of immune cells and cancer cells is not just prognostic, but also predictive of response. We've shown that it's predictive of response to immunotherapy, in non-small cell lung cancer patients treated with these checkpoint inhibitors. We've shown that it's also prognostic of overall survival in non-small cell lung cancer patients, and shown this across multiple different drugs. So as I'm sure all of you are aware, there are multiple different checkpoint inhibitor drugs. There's nivolumab from BMS, there's Keytruda from Merck, durvalumab, atezolizumab, a number of these agents. So we looked at 3 different agents, and across the 3 different agents, we found the same pattern, that the features of the spatial co-arrangement of the immune cells, the cancer cells, were uniformly predictive of response as well as prognostic of survival in these patients. One of the other questions that we've been trying to address is, what can we discern about the molecular patterns of these immune cells? So we know that there's a certain particular morphologic response, a certain spatial pattern that emerges with these immune cells, but what can we discern on the underlying molecular underpinning of these immune cells? And so in a study that's currently under review, we did a study with David Rim and Kurt Schalper at Yale and Vamsi Valshetty at NYU, where we got a series of adenocarcinomas of lung cancer, a particular type of lung cancer, as well as squamous cell cancers, also another type of lung cancer, and found that our computational interrogation revealed, interestingly, not just unique spatial patterns of immune cells that were prognostic in these 2 different types of lung cancer, but also that the underlying molecular features were different. So we actually did a co-registration of quantitative immunofluorescence images with respect to the H&E images. And when we co-registered the 2 different types of modalities, we found that the spatial patterns of the immune cells in squamous cell cancers had a different molecular composition compared to the immune composition or molecular composition for the immune cells or the immune patterns that we identified in the adenocarcinoma. So different spatial patterns emerged using this computational image analysis from the H&Es, but they also connected to a different molecular phenotype on the quantitative immunofluorescence images. So in other words, the relative concentrations of different immune cell families, the CD4s, the CD8s, the CD20s, were significantly different between the adenocarcinomas and the squamous cell cancers, which means, and why is this important? Well, it means that if you're going to treat these patients with checkpoint inhibitors, you're going to treat them, or you're trying to prognosticate outcome, you're going to take a different approach. You actually have to look at them quite differently and come up with different predictors for outcome and treatment response. Just a few different examples here in other indications. This is work we're doing in ovarian cancer, similarly having shown that the spatial arrangement of immune cells predict overall survival in ovarian cancer. We've now demonstrated in a large cohort of patients that this spatial arrangement is also predictive of survival in endometrial and cervical cancers. Just like we showed in lung cancer earlier this year at ASCO, we demonstrated that these spatial immune features were also prognostic of survival and response in different gynecologic cancers that have been treated with checkpoint inhibitors. In head and neck cancers, again, similarly have demonstrated that the spatial co-arrangement feature is prognostic of overall survival and event-free survival. What we also did in oropharyngeal cancers was we coupled the immune cell arrangement with another hallmark of poor prognosis in head and neck cancers called multinucleation. Now we know that multinucleation is a poor prognostic hallmark in these tumors, but it's very difficult to visually assess and visually identify. But what we did was train a machine algorithm to go and identify the multinucleation, create an index based on the multinucleation. And when we combine the multinucleation index with the spatial immune patterns of the TILs, the combination turned out to perform better than the individual biomarkers. Not only was it prognostic across all patients with P16 positive head and neck cancer, it was also prognostic in the individual different AJCC 8th edition staging criteria. So within the individual stage groups, it was prognostic, and that's really critical because that has treatment implications. Because right now, the status quo is to treat based on your staging from the AJCC criteria. And the fact that we can prognosticate good versus bad tumors within individual stages means that we could potentially identify patients who might benefit from escalation of therapy. So in stage 1, we've identified a subset of patients who probably need to be treated more like stage 2s, and therefore need potentially escalation of radiation therapy. On the other hand, being able to identify a subset of patients who do relatively well in stage 2 means that they could potentially be candidates for de-escalation of therapy. Skip over this in the interest of time. This is an example where we've been studying patterns using computational image analysis between immune patterns of COVID-19 patients who had died because of the disease versus patients who had died because of H1N1. In work that's still under review, we demonstrated that the computational image analysis revealed very uniquely different immune patterns in the autopsy tissue images from the two types of influenza. And so we found that there were typically much larger clusters of immune cells that typically emerge in H1N1 versus much smaller fragmented immune cell clusters that emerged in COVID-19 patients who had died. The other question we've also been asking is, where do you interrogate? So all the analysis that I've reported so far has been largely limited to the tumor. But in a study we published a few years ago, we've shown that actually there might be data that resides outside the tumor that might be very important from a prognostic perspective. So in other words, we've shown that if you look at the regions outside the tumor and the proximity of the tumor, the features of the individual cells, the architecture of the cells, actually is potentially even more prognostic compared to the features of patterns that are coming from within the tumor region alone. In this time of sort of social unrest and racial inequity, one of the questions we've been asking is, well, what can AI do to address questions around health disparity and racial equity? One of the questions that we wanted to try to understand with AI was to try to get at the fact that, try to understand the question as to why prostate cancers tend to be more aggressive in African American men compared to Caucasian American men. So in a study that we published earlier this year, we actually looked at a cohort of men who had surgery for prostate cancer who were African American versus a cohort of men who had surgery for prostate cancer but were Caucasian American, and found that when we looked outside the tumor, when we looked at the stroma, that with our tools, we were able to find significant differences in the nuclear arrangement and nuclear architecture in the stroma between Black men with prostate cancer and white men with prostate cancer. And this was significant because it sets the stage now to try to think about creating population-specific models. One of the things we know about prostate cancer and risk calculators is that the majority of risk calculators that exist today have not been trained on a very heterogeneous set of data. The African Americans, Hispanic Americans have not really been represented in a lot of these risk calculators. And so our data showed that if you create population-specific models, that the ability to prognosticate risk of recurrence in African American men went significantly up compared to a model that didn't really account for population-specific differences. So this is an opportunity where AI could potentially play a role in discovery, really enhance and extend our understanding of differences across different populations, and try to sort of potentially combat some of the implicit biases that go into creation of these models. Just a quick mention, we've also found similar differences now in studies of breast cancer, early stage breast cancer, between South Asian women with breast cancer and North American women with breast cancer, again suggesting the need for population-specific models to be able to prognosticate cancer recurrence and outcome. I want to very quickly in the last few minutes switch over from pathology to radiology. This is an active area of interest in our group. It's about 50% of our group works in pathology, the other half is working in radiology. And one of the questions that we've been asking is, how can we better distinguish on an MRI scan a treatment confounder versus a true example of disease recurrence? So this is work that's actually being led by Dr. Pallavi Tiwari, an assistant professor in our center and in biomedical engineering at Case. And she and her group have been trying to address the question of, are there differences between radiation necrosis, which is an effect of chemoradiation in brain tumor patients, versus true cancer recurrence in brain tumors? And so after patients with GBMs, brain tumors, get treated with chemoradiation, they come back for a follow-up MRI scan, and very often something shows up on the MRI scan. The question then is, like I said, is this radiation necrosis, is this cancer recurrence? very often you can't tell, so you end up having to do an intracranial biopsy. These are risky, these are expensive, and it's traumatic for the patient who's just undergone treatment for a brain tumor. We developed a new set of features called Collagen. Essentially, it's a new radiomic feature that allows for characterization of the orientation of edges on a pixel by pixel basis. So specifically, what we're doing is trying at every pixel to identify the dominant edge, and then coming up with a way of capturing the entropy with respect to the orientations of the dominant edge at every pixel. And when you do that, you're able to convert the original images into color maps like these, where essentially these color maps represent the breakdown in the morphology within specific regions. So what you're seeing with these the reds here essentially represent areas where you see high entropy, representing significantly high morphologic heterogeneity. The blues represent areas where you've got relatively low heterogeneity, more homogeneity. And so really no surprise there that, well, the cancer turns out to have high entropy, high heterogeneity. Radiation necrosis tends to have much lower heterogeneity. And so even though these differences weren't obvious on the original MRI scan, by switching it over to this entropy map, the differences become more apparent, and now potentially have the ability to obviate the need for intracranial biopsies in patients who have radiation necrosis. Just keeping up with this sort of idea of my talk of new biomarkers, right? Intuitive features, handcrafted features. One of the other things we've also been exploring in the context of prostate cancer on an MRI scan. I talked about work that we've done on pathology, on images, tissue images, but on MRI scans, we wanted to ask the question of whether there were differences in the shape of the prostate and the individual prostate compartments between patients who had recurrence versus patients who did not. And so we did a study where we took a number of patients with their MRIs of the prostate where they had recurrence after surgery, and another set of patients who did not have recurrence after surgery. We created two dedicated atlases by mapping all of the MRIs into a single canonical representation, and then did a difference atlas between the two populations, and found that there were significant differences in the distension of the capsule and the individual compartments of the prostate between patients who did have recurrence versus patients who did not have recurrence. And so interestingly, suggesting that features relating to the distension of the capsule, if you can quantify that, potentially could be another biomarker suggesting which patients are likely to need aggressive therapy versus patients who potentially could be candidates for active surveillance or watchful waiting, in which case they may not actually need to get surgery. I talked previously about looking outside the tumor, and that's sort of a theme that we found keeps coming up over and over again. And we've explored the same theme in radiology images as well. This is work that was published by Nate Brayman, a grad student in our group, just graduated over the summer. And Nate, in his PhD thesis, focused on looking at entropy features, radiomic features outside the tumor. His specific focus was on predicting response to neoadjuvant chemotherapy in breast cancer patients. And he found that, interestingly, that when you looked at patients receiving neoadjuvant chemotherapy, the radiomic features outside the tumor predicted which patients would go on to have complete pathologic response versus patients who would not have a complete pathologic response. And I know this is an area that Savannah Patridge at UW is also very interested in, and we've had some conversations about taking this forward. But this was interesting because it really suggested that the features or the areas outside the tumor could play a significant prognostic and predictive role in response to neoadjuvant chemotherapy. But like I said, this is an area that keeps coming up. There are other areas where we found that features outside the tumor seem to be very, very important. This is an example where we've been predicting and monitoring response to immunotherapy in non-small cell lung cancer. This is a multi-site study that we published late last year in Cancer Immunology Research, where we found that features inside, but also entropy features outside the tumor were implicated in predicting not just response to immunotherapy, but also survival in these patients. And what was interesting was we were able to connect the radiology with the pathology. We were able to demonstrate that the features that we were seeing outside the tumor were strongly associated with the spatial patterns of the immune cells on the corresponding biopsy images from these patients. So not only that were we able to identify a set of features on the radiology images, by connecting it to the pathology, we provided further interpretability and intuition about the meaning of these features. Very finally, one very last biomarker that I want to leave you with, something I'm really excited about, is looking at vascular tortuosity. So this is, I think, an area that's been getting some attention in the ophthalmology space. We started to look at tumor morphology, or specifically vascular morphology of tumors a few years ago, where we started to look to understand whether there were differences in the appearance and the morphology of the vasculature relating to tumors that did respond to chemotherapy versus those that did not. And we found, interestingly, that in patients who did respond, patients who had pathologic complete response, the vessels were much smoother compared to those tumors that did not respond to chemotherapy. And so we were able to convert the tortuosity of the vessels into a biomarker that allowed us to predict response, not just to chemotherapy, but as you can see on this slide, also serves potentially as a biomarker for predicting response to checkpoint inhibitors. So we've demonstrated in a number of abstracts, we currently have a paper that's under preparation, that you can really separate out, distinguish responders from non-responders to immunotherapy. We found that responders tend to have a much smoother vasculature. Again, non-responders had a much more convoluted, more twisted vasculature. And then finally, I just want to end with just a shout out to some amazing technology developed at UW, thanks to an amazing collaboration with Dr. Jonathan Liu in mechanical engineering. Jonathan and I connected a couple of years ago, and since then been having a lot of fun. I certainly have in taking the approaches we've developed in 2D pathology and applying it to 3D light sheet microscopy. This is some of our early data looking at 3D pathomics or 3D approaches to capturing the shape and appearance and arrangement of glands on prostate biopsies. Amazing technology developed by Jonathan and his collaborators, really allowing for a 3D visualization of the structure and the morphology of these biopsies in 3D. I mean, just providing unprecedented views, things that you can't possibly capture in 2D. And so our initial data suggests that the 3D features that we're now trying out from these light sheet microscopy volumes provides a better ability to characterize disease compared to the corresponding features from 2D images alone. So with that, I will end. So hopefully what I've conveyed is that computational analytics with routine imaging can help address questions in precision medicine. One of the key ingredients here is the fact that the computational diagnostics are relatively low cost. It's completely non-destructive of tissue. It fits into the clinical workflow and potentially has the opportunity for global impact, especially in low and middle income countries, because everything that I've talked about today, except perhaps for the light sheet microscopy, is really coming off standard clinically deployed technology. We're talking about CT scans or MRI scans or HD images. So nothing fancy here. I will make the argument that for AI ultimately to get clinically adopted, particularly for precision medicine, precision oncology applications, demonstrating and really establishing the morphologic and molecular basis of the imaging features becomes really important, really critical. And so I can't overstate the importance of an intuitive basis to drive clinical adoption. Like I said at the onset, this is the work that I've showcased today is really a testimony to the amazing students, trainees, postdocs, faculty, staff that I have the pleasure and honor of working with on a daily basis. So everything I presented is really their work. I just happen to be the conductor of an orchestra and just playing the wonderful symphony that they help to create. And of course, you always have to thank the funding agencies. Very grateful to the NCI, the DoD, and other foundations for making this work possible. Thank you.
Great, thank you so much, Anant. That was a wonderful talk. We have quite a few questions in the chat, so I think we'll start with those, and then if there are other questions in person that people would like to ask in person, then we can do those afterwards. All right. So,
so I think there's a question by from Larry, Dr. True. So I actually prepared for that. So so I threw it in. So I think the question. Let me just perhaps repeat the question here. So the question is about pre-analytic variances. There are multiple different sources of variation in the preparation of the slides. And so how do these tools really address that and address these issues? So we've actually, we totally agree. this is a major issue. One of the things that we've realized is that when you have different labs, the preparation, the creation of these slides can all be very different. Your scanners are different, as I explained with the example of the endomyocardial biopsies. And so what we've done is taken a two-pronged approach. One thing that we've done is taken a very sort of pragmatic, practical approach to building our classifiers. So rather than solely focusing on taking the best, most discriminating features and putting it into our classifier, we've actually said, no, let's look at how reproducible the features are. So one of the things that we do on a regular basis for all our studies is make sure that when we develop our classifiers, we make sure to include data from multiple sites. I cannot overstate the importance of this because, like Larry pointed out in his question, that is a significant source of variation in the appearance of these images. Now, there are things that you can do, for instance, color normalization, color calibration that you can do post hoc. But what we've done is that in spite of applying all of those harmonization strategies, you still have an issue that some features are going to be more sensitive to some of the pre-analytic sources of variance. So we've done a very sort of careful, meticulous interrogation of the reproducibility of the features. We try to find the set of features that really hit that sweet spot, that really maximize the reproducibility across sites, but at the same time are highly discriminating. And so the ability to find the set of features that occupy that sweet spot, those are the ones that are then included in the machine learning classifier. And the idea here, we've actually demonstrated in a few papers now, both on the pathomics side, but also on the radiology side, that taking this kind of approach really confers more generalizability. So when you validate the approach on external validation sets, these classifiers tend to be more robust, more resilient, because they've really been identified as a subset of features that really can handle the sources of variation across the different sites. So that's one thing that we're doing. The other thing that we've done is also instituted a careful quality control into the construction of our classifiers. So HistoQC, this is by the way a completely freely available program, which I encourage anyone interested to go and grab from the GitHub link listed on the bottom of the screen. This is developed by Dr. Andrew Janowick, who's a research faculty member in our group. And the nice thing about this program is that it allows you to ingest a whole bunch of slide images, and then spits out a series of quality metrics that tell you about the computational usability of the slide. And so if the slides are compromised, if they're if there's blurriness, if there are bubbles, if there are ink markings on the slide, it identifies those artifacts and sort of separates them out. So you can do an analysis of a batch, identify what slides potentially are compromised that you don't want to use for training or classifier. You can actually cull those out. Maybe you want to cull them out because they are a good test set, because they can really provide a way to validate robustly your AI tool. So just a sort of a long answer to an important question. And so these are the couple of strategies that we've instituted in the development of our approaches for addressing some of these pre-analytic